My name is Marc Cubeta. I'm a professor in the Department of Entomology and Plant Pathology at North Carolina State University in the College of Agriculture and Life Science. My area of interest in microbiome sciences is to study the interaction of microorganisms, mostly beneficial and disease-causing microorganisms that are associated with plants, and how these organisms interact with various factors in the soil, such as soil chemistry and soil physical properties. We like to try to identify novel or naturally occurring beneficial microorganisms that can be used in a context of suppressing plant diseases or insects or possibly increasing the yield. For example, we work with potato, we work with rice, we work with various fruit crops such as blueberries and apples, and we even work with other crops that are very important in the ornamentals industry, boxwoods and calibrachoa. The boxwood and the calibrachoa are projects that involve recently discovered diseases. So there's some diseases that are attacking these plants which were not known to exist in the United States before 2011. So we've been trying to better understand some of the fungi that are causing the diseases. Now we're trying to work on methods to manage these diseases that involve the use of beneficial microorganisms. The tools that I use in my research basically deploy classical microbiology and also molecular biology based approaches where we look at RNA and DNA. But more recently we've been using analytical tools that have been developed by Dr. Carbone's group to better analyze and also identify particularly unknown fungi, because that's a very challenging area of science right now.
My name is Ignazio Carbone. I'm a professor in the Department of Entomology and Plant Pathology. One of the most powerful ways to understand microbial diversity is to reconstruct their evolutionary history. And behind me here I show you one of these phylogenies that captures 1,000 species of fungi. And all around the tips are the species. And here I could zoom in on one of them, for example. And within each of these species is a lot of diversity that we're not capturing right now in this tree. And this diversity is important because it tells us something of the process of how these microbes are changing. Because this is a static tree right now, but it's really dynamic when you drill down into each species. There's genomes associated with these microbes, and we could look at variants across all these genomes to capture the dynamic of what's happening when these are species or these populations of individuals are evolving. And for example here, there are phylogenies for each chromosome, and we can actually target any single individual in a population and see how its genetic background is changing through time and space. And these rings around the tree show us how the genetic background for each individual is related to the other. And you could see what is emerging is there's two dominant genetic backgrounds on all the chromosomes. And overall, the chromosomes, we could discern these two distinct lineages. So it's not just that single species that I pointed out to you that's important, it's this resolution on a population scale within that species that tells us that we need to pay attention to these two lineages and incorporate them into whenever we're developing biopesticides or trying to manage the disease. It's this knowledge of the genetic background of microbes and how they're changing on a relatively recent time scale that helps us or elucidates better approaches to manage them in fields where they cause damage, or even to leverage them to enhance plant growth and plant yields.